Y como ha sido anunciado, a continuación escucharemos la conferencia del Dr. Drew Weissman, Premio Nobel de Medicina 2023.
I'm highly honored to be here and to be given this honorary membership to the Mexican Medical Society. It's a great honor, and thank you. So what I wanted to talk to you about today was where we're going with vaccines, what new directions, new therapeutics we're developing. I always start by showing a timeline of RNA. And I do this because when the pandemic first started and the vaccine was first released, I spent a lot of time talking to lay audiences, evenings, weekends, in person, on Zoom, all sorts of different crowds. And I was talking to them because they were nervous, they were unsure. One thing that they always said to me was they were nervous about the vaccine because it was invented in 10 months. And I always had to stop myself from laughing because mRNA was not invented in 10 months. It was first identified in 1961. It was first injected into animals in 1990 and into humans in the late 90s. So it's been in clinical trials since the 90s. It is not a new development. The background of RNA was that after the human clinical trials, these were an unusual vaccine. What they would do is they would take dendritic cells out of a patient, pulse them with different kinds of RNA, and then inject the dendritic cells back into the patient. They did this because if they injected RNA into a person, the person got sick. We now know the reason why that happened. There are 17 different innate immune sensors that recognize RNA and make inflammatory cytokines, type 1 interferons, directly inhibit translation, induce apoptosis, and cell death. So the work that Katie and I did was try to understand why RNA was so inflammatory. And this was the telling experiment. What we did here is we isolated RNA from both mammalian cells and bacteria from different fractions and different organelles and tested them all for inflammatory activity. The thinking was if all of them were inflammatory, then there was no solution. But that's not what we found. What we found was that tRNAs were least inflammatory and bacterial RNAs were the most. That led to the hypothesis that maybe nucleoside modification was responsible for the inflammatory activity of the mRNA. Transfer RNAs have up to 25% of their nucleosides modified, and bacterial RNA has very few. But there's over 100 different types of nucleoside modification, and they're conserved throughout evolution, and M6A was shown to be reversible. So what Katie and I had to do was we had to make nucleoside-modified mRNA, which had never been done before. The way our bodies do it is with an enzyme and a short RNA, and the RNA identifies a sequence where the modification is placed. That wouldn't work for an mRNA where we wanted diffuse and many different modifications. So what we did is we used the standard phage T7 RNA polymerase, and we tricked it into incorporating a modified nucleoside triphosphate. And you can see from all of these RNAs, these are what we were able to make. And what was interesting when we tested their inflammatory activity was that any uridine modification reduced the immunogenicity, the inflammatory activity of the RNA. But the next experiment was more exciting. Here we tested, could the modified RNA still be translated? And we had made a bet the evening before we did this experiment that it wouldn't be. We were wrong. And we were wrong tenfold in the wrong direction. Not only was it translated, but it was translated much better. We also found that the phage polymerase made contaminants in the RNA that also reduced inflammation of double-stranded RNA. So we figured out how to purify those away. And when we combine nucleoside modification with purification of double-stranded RNA contaminants, we could increase the translational effectiveness by 4 logs, a huge increase in translation. We then had to figure out how to deliver the mRNA. Just about every fluid and cell in our bodies contains RNases, enzymes that degrade single-stranded RNA. So we looked at about 50 different formulations and finally tried lipid nanoparticles. Lipid nanoparticles, or LNPs, were developed to deliver short interfering RNA. They're in FDA-approved drugs and are used in patients today. But siRNAs are 21 base pair double-stranded RNAs. mRNAs are 2 to 12, 14, 16,000 single-stranded nucleotides long. So we had to figure out, could we incorporate an mRNA in an LNP? To do that, we use luciferase encoding mRNA. Luciferase is an enzyme in fireflies that produces light. So you can measure translation by light in the animal. And what we found is that any route we used to deliver the mRNA resulted in translation of the RNA. But what was most striking is that peripheral administrations, ID, IM, sub-Q, resulted in long-term 10-day translation of the RNA. So when you think about vaccines, the important things are listed here, and I won't go through all of these, but the important thing to note is that the best vaccines we have are live virus, attenuated virus vaccines. And the reason they work so well is they infect the cells of the host and they produce protein for a long time. For a vaccine to be effective, the longer the duration of protein production, the better. So now we had an mRNA that made protein for 10 days and didn't require a live virus to do it. So we worked and we started developing modified nucleoside vaccines. And this is probably around 2013, 2014. I'll quickly go through this data, but what I'm trying to show you here is the comparison of influenza vaccines. And the inactivated virus is what us old folks get. The live virus is what our young children get. The mRNA-LNPs were 50 times higher than inactivated virus and 5 times higher than live virus. That caught people's, and especially biotech companies' attention. We wanted to understand why we had such a potent response. So this is the cartoon of how B cells respond. And what we did is we measured different of these B cells in an antigen-specific manner. So here we're counting the number of B cells that recognize influenza HA. In the middle is inactivated virus. And what you can see is that there's 50 times more antigen-specific B cells of all 3 phenotypes, which explained why the antibody titers were so high. We forgot about some mice. They sat in our colony for about 13 months. We paid for them, we just didn't realize they were still there. But we finally found them, and we tested their bone marrow, and we measured antigen-specific long-lived plasma cells. And what we found was there was an enormous number of HA-specific plasma cells in the bone marrow. But we had a problem. So I showed you early on that the mRNA had no inflammatory activity. The LNPs, as far as we knew, also didn't activate innate immune sensors. So we couldn't figure out where the adjuvant was in this vaccine. Adjuvants are required for an effective vaccine to work. Adjuvants deliver inflammation, and they activate different kinds of T cells. Typically, adjuvants are Th1 or Th2, which is cell-mediated immunity versus systemic antibody, but there are many kinds of T cells. And we focused on a population known as T follicular helper cells. These are cells that form the germinal center and interact with germinal center B cells where they stimulate antibody responses. We used the macaque model system and used an HIV antigen. And in the middle, this is a comparison to the identical envelope protein with double-stranded RNA adjuvant versus the mRNA-LNPs. And we're measuring antigen-specific T follicular helper cells. Double-stranded RNA was thought to be the best adjuvant for inducing TFHs. But as you can see, the mRNA-LNPs were much better. We calculated that about 50% of the T cell response were TFHs. But then we had another problem. We didn't know where the adjuvant was. It wasn't in the RNA. It wasn't supposed to be in the LNPs. So we did a simple experiment. We took empty LNPs, so lipid nanoparticles with no RNA, and added them to influenza HA protein. And you can see in red and green, the green is empty LNPs with protein, and the red is mRNA LNPs with the same antigen, and both of them induce TFHs. So the lipid nanoparticles are the adjuvant that specifically induced TFHs, which hadn't been described for any other adjuvants. When we looked at the molecular mechanisms, what we saw was interesting. The LNPs induced IL-6. In mice, IL-6 induces TFHs and didn't induce type 1 interferons that inhibit TFHs. So it seemed like they were a designed adjuvant that we found totally by luck that induced potent antibody responses. I'm going to jump ahead just because it's getting late. We've also worked on a variety of HIV antigens. And in this case, what we're doing is we've designed new ways to make viral-like particles. In a vaccine, viral-like particles are very potent antigens. The HPV vaccine is so potent because it makes a viral-like particle. Hepatitis B makes a viral-like particle, both incredibly potent vaccines. Tetanus toxoid is a protein, it's not very potent. So we designed a number of different ways to make viral-like particles using different hypotheses and approaches. And then we immunized macaques with these different VLP-producing envelope immunogens. And what you need to note here is that the middle antigen, VLP, is making autologous neutralizing antibodies after a prime. That's never seen in HIV. and it's making titers in the hundreds of thousands after a boost. So the combination of a VLP with modified RNA-LNPs is inducing incredibly potent HIV vaccines. And these are in clinical trials right now. And we're hoping to find potentially a new vaccine to prevent HIV and even to treat HIV in the future. So I wanted to end by talking about a new vaccine that's directed against a bacterial pathogen. RNA vaccines are essentially designed for viruses. Viruses infect a cell and make their proteins. The LNPs transfect the cell and make proteins. So they present responses that effectively target viruses. Bacteria are very different. Most bacterial vaccines are carbohydrates because the bacteria have a thick coat covered in carbohydrates. And what's more difficult is when you want to deliver a bacterial antigen as an RNA, bacteria don't glycosylate their proteins, but mammals do. So now you have to worry about glycosylating bacterial antigens. So it's a very complicated process to make a bacterial vaccine. So we, of course, started with one of the most difficult ones, C. difficile. As you know, C. difficile is a leading cause of in-hospital infections, but it's also now become a community-acquired disease. It has enormous burden on the hospital itself, on the economy, and on humanity. We chose a number of immunogens. I'm going to show you the first three. So a few other companies have made C. difficile vaccines. They include toxin A and toxin B. None of those vaccines have made it through phase 3 clinical trials. The likely reason is that they don't get rid of colonization. They simply treat the acute infection, but the bacteria stays behind. And as you know, 30% of people who develop C. diff develop recurrences, and sometimes multiple recurrences. So we added an additional antigen, and this is an antigen associated with the cell surface of the bacteria and spores. We developed a number of different RNAs, and we immunized mice, in this case 2 times. And we measured the titers of IgG. We're comparing these to the identical proteins adjuvanted with alum and CpG. And as you can see, the RNA vaccines are 5 plus or minus fold better than the protein vaccines. The number of antigen-specific cells, again, was very high. Most importantly is when we did the testing. So we didn't do an easy test. We gave these animals 50 doses, 50 toxic doses of toxins. And the RNAs completely protected the animals. The proteins partially did. We then challenged the mice with C. diff spores and asked, could the vaccinated mice survive? And what you can see in the Kaplan-Meier on the left, the vaccinated mice lost a tiny little bit of weight, but quickly gained it back and survived, whereas the unchallenged mice died. So the vaccine was effective at protecting mice from C. diff infection. The clinical scores followed the weight loss. The most important experiment, though, is when we followed the level of toxins in the GI tract of these mice. And what you can see, so the green is bivalent, it's the two toxins mixed together. The purple is the trivalent with the outer coat protein. And if you look at the later days, the purple goes to 0, goes to limit of detection. That means that we've cleared the bacteria from the mouse's gut. And we went back and we cultured the GI tract of these animals, and the C. diff was gone. So not only were we able to treat the acute infection, but we were able to decolonize the animals. So we're now working with a company to develop this vaccine for moving forward. And I think given the time, I will stop there. Thank you.
Señoras y señores, a continuación el doctor Raúl Carrillo, presidente de la Academia Nacional de Medicina de México, hará entrega al doctor Drew Weissman del diploma correspondiente por la conferencia dictada. Muchísimas gracias a nuestro conferencista invitado, el doctor Weissman.